Hey everyone, and thanks for joining me today for episode 853 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and I'm excited to be speaking today with our guest, Dr. Dawn Smith. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And today, Don is going to share some of his insights and stories with all of us. So listeners, get ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Donald Smith. So, Don, welcome to our show today. How are you?
I think I'm quite all right. Up here in Canada, it's just begun winter, so holding my breath for spring.
I understand that. I'm not a winter person myself, so... Well, we're excited to learn more about you, the work that you're doing up there at McGill, and all of the great things that you've done over the years. But before we go into those details, let me take a moment to tell our listeners a little bit more about you and how you got to where you are today. So listeners, Don is the Distinguished James McGill Professor in the Department of Plant Science at McGill University. He's also CEO of BioFuelNet Canada, head of Biomass Canada, and head of the McGill Network for Innovation on Biofuels and Bioproduct. Don received his bachelor's and master's degrees from Acadia University and his PhD from the University of Guelph. Afterwards, he worked as a National Sciences and Engineering Research Council of Canada, or NSERC, Postdoctoral Fellow at Agriculture Canada before joining the faculty there at McGill in 1985. Over the course of his career, Don has received many awards and honors, including the Clean 50 Award for contributions to sustainable development and clean capitalism in Canada, and the Queen Elizabeth II Diamond Jubilee Medal, in this case for significant contributions to intelligent agriculture. He's also a fellow of the Canadian Society of Agronomy, and he served as a member of the Canada-US think tank on climate change and agriculture in northeastern North America. And Don, today we're excited to get to know you as a scientist, but also as a person more broadly. So can you tell us, what do you like to do when you're not doing science?
I walk. I like to read novels, things like this, a little bit of escapism. I like to travel. I really like to travel, and I often manage to travel in combination with work.
Perfect. And you mentioned reading. I love to read myself. Do you have a favorite book or a book recommendation that you'd like to share with me and listeners today, whether it's related to science or not?
There are many, but one of the ones that had the biggest impact on me over the last several decades was Gravity's Rainbow. It's a sort of sci-fi and pretty out of left field, but it's an interesting book. Lots of thought-provoking stuff in it.
Very cool. So what about this particular book had a big impact on you?
It's set near the end of the Second World War, and the use of missiles is just beginning. So it's kind of a turning point in everything else that's going on in the world with humanity. And there's just a lot of interesting perspectives from a lot of different people, and that makes it quite interesting as well. But if you know the author, Thomas Pynchon, he always takes a very off-the-wall perspective on how people view things.
Interesting. Well, we will add this book to our website for our listeners to find. It's wonderful to hear a little bit more about what you do when you're not working in the lab, reading, getting out there, traveling, and even just going for a nice walk is a great way to clear your head during a busy day. So I love that. But how do you describe your work and what you do in the lab to people who might not be familiar with your specific field or might be outside of science altogether?
So I started here 4 years ago this year. At that time, I came from Southern Ontario, which is as far south as you can get in Canada. And I've been working on soybean there. So when I came here, they had just developed soybean types that were possible to be grown in Quebec, where I am now, so the Western Quebec, because they could actually mature quickly enough to get the seeds produced, et cetera, before the frost could. So we started growing them, and we noticed it was kind of odd. They'd come out of the ground. They look nice and dark green. These are legumes, so they fix nitrogen. They take nitrogen out of the air. They don't need nitrogen as fertilizer. So this wasn't surprising. They were nice and dark, and they have a high protein level, part of the reason we value them. But anyway, they were nice and dark green. And after a short time, they would start to turn paler green, and some of them would go almost all the way to yellow. And we were watching this for the first 2 years, and I was going, what is going on here? Because eventually they would suddenly darken up and then go dark green again, and they just carry on. But the first few weeks of their getting established as a crop they looked very nitrogen limited. This is not a good thing. So I was looking at it carefully. And the thing about soybean is that it evolved in the tropics and subtropics, mostly the subtropics, south coastal China, et cetera. And it was domesticated there. And it's all set up for high temperatures. It forms these nodules on its roots, and the bacteria live inside them. And the bacteria take nitrogen out of the atmosphere and feed to the plant, which means you don't have to give them nitrogen fertilizer, which is one of the most expensive inputs for crop production usually. So this is a nice thing about legumes in general, but it just seemed that it was not happening well in Quebec. And so we began to wonder if this could be related to soil temperature. So I did a bunch of checking. It turns out that the soil temperature, the optimal soil temperature for nodulation of soybean had been known for over 100 years. And it's actually in the 25 to 30 degrees centigrade range, which is warm. And that's certainly well above what we were getting here in the spring. At seeding time here is often 10 degrees centigrade. So that's a lot cooler. So we did some testing in the lab. We were able to show that low soil temperature was causing the problem. We did some testing to see if all stages of the establishment of this symbiosis, the nodulation, all that kind of stuff, were equally sensitive to it or just some parts. And it turns out whatever was going on in the first 12 hours was particularly sensitive. So we did some testing. It turned out what's going on in the first 12 hours, we're lucky. Some other people were just publishing it. So in the first 12 hours, the two partners, the plant and the bacteria that will eventually go inside the nodules, they're kind of doing a chemical handshake. They're exchanging signal compounds. The plant's producing these phenolic compounds, isoflavonoids, genistein, et cetera. And these compounds turn on a bunch of genes in the bacteria, and the bacteria then produces a return compound, which is a lipocyto-oligosaccharide. I tell my students sometimes if they've been out drinking the night before, they come into the lab the next day and they can still say that word, they can stay in the lab. So we did some testing. What we did is we took the genistein that the soybean plant produces, we put it into the culture of bacteria in the evening, just before we went home at the end of the workday. And we just left it sit overnight in the lab. So we come in the morning, we take that liquid culture to the field and use it as an inoculant for our soybean. So by this time, all of the signaling stuff from the plant to the bacterium and the bacterium turning on its genes and making the return signal had all happened at room temperature. In centigrade, this is like 20, 21 degrees. So it was all done. So we tried it, and it really helped things move along quickly in the field in terms of the nitrogen fixation. It sped things up quite a lot. We got increases of up to 80% in total seasonal nitrogen fixation. At the same time, this is like the middle to late 90s. We were going through a recession, and grant funding was getting harder to get. I had a really good technician. I was where I would be able to pay him. So I looked at this. I said, okay, I went and talked to the guys here at McGill. I said, could we patent this? This resulted in me learning a lot about patenting and all this kind of stuff. I'd never done it before. So but they said, yeah, potentially. So they did some checking and they said, yeah, you could. So we patented it, and we started a company based on the patent. And my technician was the president of the company. So he had a job. The company was then doing testing at field sites over the next few years. And we noticed several years in a row that the plants definitely didn't go through anything like the period of nitrogen stress they had been going through before. But the other thing that we noticed that we didn't expect was that the plants came out of the ground faster. The seedlings came out of the ground faster. And that happens well before the nitrogen fixation starts. So we're puzzled by that. The first year, actually, I brushed it off. I said, oh, that can't be right. But the next year, I was going, maybe. I had a grad student who had seen it at several sites and took data on it. So you go, okay. We looked at that. We said, well, what could be going on here? It was only happening at the sites where we had added the genistein into the inoculum. So it was producing the lipocyto-oligosaccharide return signals. So we tested the genistein, the bacteria by itself, the bacteria plus the genistein, which made the LCO because you couldn't buy the LCO at the time. We tested these things to see if it was one of those, maybe just one of them, that was causing this increased growth. And it turned out it was the LCO, the lipocyto-oligosaccharide. So we patented that, put it into the company. But this was kind of a bigger deal because the interesting thing was with the signal molecules in the nitrogen fixation symbiosis, they're very specific. It has to be the right isoflavonoid out from the plant and the right lipocyto-oligosaccharide back of the plant. And they have to be correct. With the stuff we were seeing, any LCO would work. It didn't care, and it worked across all plants, which suggests that the use of LCOs for making the plants grow better was probably evolutionarily older thing. It's easy to go from the general to the specific. It's hard to go from the specific to the general. So that was the thing about that. It looked pretty nice in many, many ways. And we did commercialize it. Then the company was moving along. The company was eventually, it went through what I like to refer to as a corporate food chain. It was eventually taken up by a larger Canadian company. They moved their technologies closer to market. Then they sold themselves to what was the largest distributor of inoculants in the US at the time, a company called EMD. I think, if I remember correctly, it was for 32 million euros.
Oh, wow.
EMD had it. Then EMD sold a part of themselves to a company called Novozymes. Novozymes hardwired themselves to the dreaded Monsanto, and Monsanto was swallowed whole by Bayer. Myself, actually, and one of my current industrial partners were talking to the guy from Bayer who had just retired from a company about 6 years ago. But he was in the right part of the company, so he had some dealings with our technology. He told us that Bayer had made about $1 billion on it then, so maybe $2 billion by now. For quite a number of years, McGill was getting more in royalties off that patent than all the rest of their patents and technologies combined. So it was kind of interesting in all that sense. And now we're working on trying to look at a whole series of other plant-associated microbes and looking for similar signals and finding relationships like that. That's where we are now.
Well, Don, this is a remarkable story. And I guess if you had to summarize your work in maybe a sentence or two, how do you describe this large body of work you've been involved in over the years?
Well, we just say that what we've discovered is the large group of microbes that live in association with plants, various structures in the plants, but the biggest communities in the roots. That's not new. We didn't discover that. But what we discovered is that a lot of the stuff going on between the microbes and the plants, they've been co-evolving for probably 500 million years. And there's a lot of very subtle stuff. And so the plants are at least often probably signaling to the microbes. The microbes are certainly signaling back, and we've been able to isolate a number of microbes and we're able to show that they're producing signals that are causing plants to grow better. The one thing I did leave out of the other story was that when we did testing, we were able to show that the plants responded much more strongly when there was a bit of stress present. So if you're in a bit of drought stress or cold stress, the plants really respond.
Well, Don, I think this work that you're doing is so cool. And I'd love to hear a little bit about what motivates you. I think as a scientist, the work that you're doing can be really exciting, but oftentimes you hit roadblocks and just difficult periods. So do you have a favorite quote or a saying or just something that really keeps you motivated and inspired?
I'm always interested myself in the relationships between these organisms, and they're always surprising. We had never imagined that those signals would be used in the nitrogen-fixing symbiosis and also in this other relationship, and that they would be so generic. And the other thing we found out about them was that they show a bigger effect, as I said, when there's some stress present. So they seem to be, by and large, helping plants deal with stress, although there's some suggestion that a few of them, that's not exactly true. So I guess the thing I always say to my students when we're talking about what to look for and what to be surprised about, and what not to be surprised about is I'm always telling them evolution never sleeps. If there's some problem that presented, evolution will try to find a way around it. It doesn't care about any human rules on what can and can't be done. It's whatever works.
Well, I think that is a fantastic motivational quote. And I'd love to hear a little bit about some of the people who might have inspired you over the years. Don, could you tell us, do you have a particular person who you think of as maybe a role model or a mentor, or just somebody who inspired you and helped you get to where you are today?
Maybe not anybody specific, but the whole general business about science and about trying to find out how things work and how the pieces fit together. And I guess there's the process of science. I'm very grateful for the process of science. When collects data, produces new information, and you publish it, and then other people test it. And when there's a mistake, it gets corrected. And there's not many other human areas of activity where that kind of thing happens. So there's always pushing forward to expand human knowledge, but there's always testing to make sure that what's been added to the general plate of understanding is correct.
Definitely. I think just this idea of the scientific community working together to advance the field and to advance science as a whole is a beautiful part of working as a scientist.
Yeah, I quite agree.
And we talked a little bit about what you've been up to since arriving at McGill and what you've been spending your time doing over the past couple decades there. But I'd love to go back even further and hear a little bit more about your scientific origin. So, Don, can you tell us what first got you interested in science?
I've always found it interesting. When I was young, you'd see stuff on TV and things, you go, oh, that's pretty cool. But I was growing up on a farm in rural Nova Scotia, and you're in those circumstances. You spend a lot of time engaged in serious physical labor, and you don't always have as much time to think about these things as you'd like. But the whole idea about how things fit together always seemed kind of intriguing to me. And honestly, in the end, I went through, working on the farm. I had a few summer jobs while I was in high school, often blue collar laboring jobs. And there's nothing wrong with that, but it's just specifically, I had I finished my first year of university. I didn't do very well in my first year of university. I have been in the habit of skipping a lot of classes in high school. It wasn't affecting my marks, so that was okay. I started doing the same thing in university and discovered it doesn't affect your marks here.
Right. It's not okay.
It's not okay. That's right. The summer after my first year, I had a long debate with myself. Was I going to go back to university or just get a job? I was working in a fertilizer factory, and it was the middle of July, probably. was a whole bunch of phosphate came in from somewhere. It was in 100-pound bags. So there were only a few of us on the floor at that time. So we spent an entire week carrying those bags off these large 5 trucks and piling them inside. And I was used to physical labor, but it was just going on and on and on. And somewhere in the middle of that week, around Wednesday or Thursday, this is just too boring. I want to do this for the rest of my life. So I just said, okay, I'm going to go back to school. I'm going to go to all my classes, do all my assignments, and see how it goes. Right. So that's what I did. I went back, and I think at Christmas, I had A grade. Whereas I think for previous year at Christmas, I had D grade. So it was a big difference.
Well, it sounds like you really turned it around. And I think making that decision for yourself can be an empowering thing where you decide, okay, I've tried this thing. I don't like it. I want something different for my life. I'm going to make it work. And I think that is a really powerful approach. And it sounds like you were interested in, okay, I need to go to school. I need to find a different solution. What turned your eye towards science and biology?
I was growing up on an apple farm. So there were plants there and we were spraying stuff on them and things like this. And I was always a bit curious about what we were spraying and why. So when I got into biology, this sort of took my attention. I moved into that more and more, and it became what my undergrad career was more focused on. And then when I eventually went to grad school, that's what I went into. So,
and did you anticipate that you would become a professor? Did you consider industry? What, I guess, was your thought?
No. I mean, I was the first one in my family to go to university. Mm-hmm. I never imagined that I would wind up with a PhD. It just never crossed my mind. Every once in a while, even now, the other day, I was walking in the hall here. I can't remember what I was doing, but somehow I went up, I was looking around going, how did I wind up here? It's not what you would have guessed. I'm trying to think after my undergrad degree. I just didn't know what I should think about doing next. And grad school, somebody had mentioned grad school to me, but other people were, you should get a job. You have a bachelor's now. You can get a good job. Get a job, right? And I actually had one guy, when I went to grad school, he asked me what I was doing. He hadn't seen me for a while, and I told him I was in grad school. He goes, couldn't you get a job? It's like, why would you do that? Anyway, it's just all these different things. But I wasn't sure. I had been sort of doing a bit of marine biology and marine botany, and I'd spent a summer scuba diving for Natural Resources Canada in Prince Edward Island, which was interesting. We were at a place down in Egmont Bay, and they put us up in these tourist cabins, which were very minimal. And there was really nothing inside them except one book. There was one book in them. It was one of those Time Life books, and it was on Australia. I just didn't know what I was going to do next. So in the end, I decided that what I would do in the fall is I would go to Australia. Pretty random, but there you are. So, and actually it was kind of one of those interesting things. This was 1974, a long time ago. I went with another guy that I was working with at that site. We went in the last week from 1974 because the rules changed January 1st, 1975. So because I went into Australia before the rule changed, I went into Australia as an Australian citizen. I was a citizen of a Commonwealth country, so I was an Australian citizen. That was it. So it meant I was able to work and things like that. So I was there for a few months working in a steel mill. It's interesting in many ways.
Definitely. So then what got you back on the academic path?
Well, I had a really interesting year. I worked in a steel mill for a while. I ran into some young people who were traveling at the time. I wound up going up through Southeast Asia and through over to Europe and back home. But that was a long time away, and it's certainly enough distance for reflection, right? Now I was going to do something with my life. So I was going, well, maybe I'll try this grad school. So I went and did a master's. So that's how one thing led to another.
Definitely. And you went on to get your PhD there at the University of Guelph. And then how did you decide to go on to do that postdoc?
I had a summer job with a professor at the end of my master's with a professor at Dalhousie University in Halifax. And he was a bright guy. And he influenced me. He said, if you're interested in agriculture, you should go to Guelph. So I went to Guelph.
And then you did the postdoctoral fellowship at Agriculture Canada. Can you talk a little bit about that experience?
I keep telling my own grad students is just keep your eyes open. There's a lot random happens in life. You just have to seize the moment. Because I was applying for jobs near the end of my PhD. And I applied for one in Western Canada. I can't remember the name of the town now, the small town. But anyway, there was an AAFC station there. So they actually interviewed me and they actually offered me the job. But by this time, I had a partner who became my wife. She also had a PhD. And I'm looking at this small town. I'm going, there'll never be 2 jobs or PhDs here. So I actually turned it down. I took a postdoc at Ottawa instead. And the postdoc was interesting. But the most important thing to come out of it was that after I'd been there about 5 months or something like this, there was a guy turned up. He was from McGill and he was on a sabbatic year. So I got to know him. He was a nice guy. So I got to be a little bit on the sort of friend side of things. And at some point, after I don't know, a few months, he said, oh, they're going to advertise a position in our department and you should apply. So I applied and I got that job. But that was much more likely that my wife could get a job there. So that's how one thing led to another pretty randomly.
I think serendipity plays a huge part in the career path.
Really does. you cannot, I think, overestimate the role of that.
Oh, definitely. Well, it sounds like things have been going well for you there at McGill over the years. So it sounds like it's a good fit and you're enjoying your time there in the lab. But let's talk about some of the work that you're doing now. Is there a particular project that you're working on right now, Don, that you were just the most excited about and want to share with us?
We did this stuff with the lipocytoligosaccharides. Then we did it again on a somewhat larger scale, still pretty small scale. We checked a bunch of strains from one of our local sites, and we isolated some strains out of that looked pretty promising. We isolated some things that they were producing that looked pretty promising. One of them turned out to be a small protein, which is known as a peptide. And that's pretty interesting. And that's starting to take off commercially now by itself. And there's really a lot to be said about that. So it has been really interesting in many ways for us. And so what we've done recently, we got funding from the federal government to carry this to a much larger scale. So we're doing some broad general screening, trying to collect a number of strains that look really promising and see if we can find a number of other signal compounds. The thing is, when you think about it, when we first isolated the signal compound from this second bacterium that looked really interesting, I had a really good master's student working on that at the time. And I just assumed it would be another lipocytoaligosaccharide type of compound. I told her that. So she went off and she did some staining just to see if it could stain it for lipocytoaligosaccharide LCOs. And it didn't stain for that. So she just tried a bunch of stuff and it lit up for protein. And I was totally surprised. I'm going, no, really protein? He goes, yep.
Try again. Yeah.
Yeah. And well, that's, always when you get a surprising result, you have to at least try it twice more just to make sure it wasn't a fluke. But yeah, she tried it and it was a protein. So we did a bunch of testing with it. So we go, okay, it's a protein. And that's all we knew. It was a protein. God knows there are a lot of proteins in the world. So she was doing testing. And she's a very bright student. I mean, she did a PhD eventually in same sort of stuff, but related to medical side of things. And she wound up running a big chunk of one of the large pharma companies in Toronto for a while. But she called me up one night about 11 o'clock. She was all excited. She was just reading this. I think I know what it is. I go, what is it? And she goes, well, it's one of these little proteins that, well, they don't have to all be little, but it's a bacteriocin. And what they do is the bacteria produce them to kill closely related bacteria. So what it does is it clears the niche space, the place where the bacteria live, of other bacteria that are reasonably closely related and so have pretty much the same resource demands, right? So they're gone. So it takes a lot of pressure off whatever resources are available for the species that's producing the toxic protein, right? They actually use these things as food preservatives. So you go, oh, that's kind of amazing. So you got this thing, it's acting as a signal to the plant to make the plant grow better, particularly when it's stressed. And it's also acting against these other bacteria. So you go, interesting, because what it's doing then is it's a dual function peptide. It's clearing the niche space of competitors. So that's good. And it's also making the niche space bigger by making the plant grow better. So it's doing two things for the bacterium. And this turned out to be interesting because we did some work with another lab and we were testing, we got a partial amino acid sequence on the peptide. So then we could guess what the nucleotide sequence might be if we're looking for genes. So then we started looking for the genes for this thing. And when we found the genes, there are 3 copies, 3 tandem repeats of the gene on a plasmid. And they'd been there for a long time, clearly, because they had evolved. So the nucleotide sequences of the 3 were at least somewhat different, right? So they've been separated for a while. But the really interesting thing about it was you have this peptide, but it's doing 2 things. So you have to be really careful in the context of evolution. If you cause an increase in one of the activities through a mutation, et cetera, but it decreases one of the other activities, that could be a problem. So it constrains the evolution quite a lot. And what we found when we looked at these 3 copies of the gene, that there were differences in the nucleotide sequence, but the differences in the nucleotide sequence were always in the 3rd codon position. So when it's in the 3rd codon, there's a number of different nucleotides that can be that spot, and they can code for different things, but there are more nucleotides than you need at that point. So some of them, you got 2 or 3 of them that code for, they actually basically say the same thing. So if either of those, say 3 of them are there, that piece still codes for the same amino acid. So they were all at the 3rd position and they were all redundant, meaning that they did not change the amino acid that was coded for. So the nucleotide sequence was evolving, but the protein wasn't.
Interesting.
Yeah, I was a bit taken aback. About the 5th time I was taken aback in this work. Looking at this going, wow. But it means that, again, because it has 2 functions, evolution is being very cautious with it. And I know that's being anthropomorphic. But anyway, the only changes in the gene sequence that were being tolerated were ones that did not change the amino acid sequence. So it was a big surprise.
Oh, absolutely. And Don, I think these surprises, these unexpected outcomes can be really invigorating and kind of make the project even more interesting than you initially thought it would be. And I know to do exciting, groundbreaking research, you need to have suppliers and partners that you can count on. So we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of ELISA kits. which offer high sensitivity, specificity, and can be used for a wide range of applications. Innovative Research ELISA kits are manufactured in their lab in Novi, Michigan, and are shipped directly to your location. They're a valuable tool for researchers and clinicians who need to detect and quantify specific biomolecules. Visit www.innov-research.com. That's www.innov-research.com to view their entire line of ELISA kits. And returning to the conversation with Don, I think listening to your story about some of the work that you've done over the years, it sounds like even though you had perhaps some surprises along the way, everything went pretty smoothly. You know, the pieces just sort of fell into place.
Well, that's how I present it now. I mean,
I was going to say, I suspect that's not the whole story.
No. There were lots of those. I mean, I've learned to embrace the surprises. You're quite right. Often it'll turn out that you get a surprise in a piece of research and it doesn't come out the way you expected. If I have that to think about a PhD student who's doing something and they don't get a response that we thought we were likely to get, then I have them do it again. And if they get the same response twice, then I say, okay, we have to sit down and think about what the heck does this mean? But it's often more interesting than what you had anticipated initially. And that's happened a bunch of times when we've gotten an answer and didn't jive with what we were expecting. And we checked it and that's what it was. So then you say, well, what is this telling us? And it often turns out that biology has done whatever. We expected to do something and it just didn't care. So we have to go off but we discover things like this small peptide. I was expecting it to be, because its effects on the plant were similar to the lipocadoliglyceride. Well, probably be similar compound, but not at all. Nothing similar at all about it. So we had lots of these interesting moments.
Absolutely. And I think in addition to these surprises, there are also just a lot of challenges and struggles and failures as part of science. So do you have an example that you could share with us of something that you really struggled with or a major failure you encountered in your career?
One of the things people don't realize is that failure is an important and actually ultimately positive part of science. For instance, when we were growing them in the field, we would often see pretty good responses to these treatments when we're doing the LCO work, right? So we put the genistein in, so we generate the LCO. We wouldn't always see it, but in the field, nothing is ever completely consistent in the field. The field environment is just so variable, but it is pretty often. And indoors in the lab, we would see it too. But about a third of the time, we'd see nothing. And I'm going, what the heck is going on here? I mean, you're seeing it often, and you go, okay, it seems to be real, but we're missing something here. So I was standing in the field one day. I was a cornfield, and it was a heavy clay, cold spring, wet. So the soil was cold. And I was looking at the corn seedlings, the corn seedlings where you had added, in this case, this is going back to the LCO, where we added the genistein, so there's LCO being produced. They were about 10 centimeters high, which we want about. So they were a decent size. In the control plots, they weren't up. There was nothing. So I'm looking at this going, this is a pretty stressful situation. These guys don't seem to care, and these guys do. And I was going, so maybe it's about stress. And we went back, and I had a really good student that just arrived in the lab at the time, a PhD student. And so I gave this problem to her, and within about 6 months, she had very clear evidence that when there was stress present, you'd see these effects really clearly. And when there wasn't, you might see a little and you might not, but you would never see more than a little. So yeah, initially in the lab, we were getting this inconsistent failure, which was just driving me crazy. And it turned out that it was telling us something really important.
Absolutely. And I think just the importance of observation here, like you were out there in the field staring at your study system, thinking like, okay, what around here could be possibly affecting it? And I think getting out there and being able to put your eyes on it can be really valuable.
Yeah. And I think in the lab, what was going on was that if we put the stuff on and the student had just watered the plants, there's no stress. But if their plants were getting to the point where they needed water, then they were stressed. So it's just things like that, you know?
Definitely. Well, thank you for sharing one of these challenges that you and your team worked through. And I know you have many success stories as well, and we'd love to celebrate some of your wins over the years. We talked about a couple of them in that conversation earlier, just about what you've been doing over the years. But do you have a favorite success story you'd like to tell us about today?
The LCO is my favorite probably, but there has been a number of it. And we also do some work on biocontrol, for instance. We had some stuff going on Clavibacter michiganensis, which is an important pathogen of tomatoes. And again, we were working with bacteria that could help control it. And we isolated a number of them, and they were very effective against it. And eventually we were able to show that the strain we'd isolated that was effective against the CMM, as we call it, was producing something, and it was diffusing away from the cells, and we were excreting it. And it was either biostatic or biotoxic. So it was either killing the CMM or making it unable to grow, either way. But I had a really good student on that too. And eventually we were able to actually isolate two compounds. We worked with the chemistry lab on this because it was just a bit more chemistry than we were used to. But yeah, we were able to isolate them. There were both isoquinolone rings. And I don't know if you're familiar with isoquinolones or quinolones. I told you earlier, I went up through Southeast Asia, right?
Yeah.
When I did that, I acquired malaria. So I had some experience with quinolines. So when it turned out to be an isoquinolone, I'm going, oh my goodness, yeah. But they work well against. So anyways, it's another interesting direction of this plant microbe interaction stuff that we were doing in the lab. It turned up some interesting outcomes. And we spent quite a long time on the structure. Not easy to do those kinds of things. And we got the structures of the two compounds almost completely. There was one side chain we weren't sure which way it was oriented. And one of them had one extra methyl group compared to the other. And we just never figured out where that methyl was. After 3 years, I said, okay, that's enough.
We're close. Yeah.
yeah. We're close enough. We'll publish it. It's okay. Yeah.
Well, this sounds like a cool project. I appreciate you sharing it with us. And one of the things that we've touched on throughout our conversation are these opportunities for travel. You mentioned at the beginning that travel is something that you particularly love. And I think you do get a lot of opportunities to go to different places as a scientist. So do you have a particular travel memory of yours that was just your absolute favorite, or a place that you've been to that's really stuck with you over the years?
I think at this point, this is about 200 countries in the world. I've been in, I think, around 110 of them.
Oh, wow.
A lot of it's kind of accidental, right? It's like you wind up going because of work you don't have any control over.
Mm-hmm.
Some of them were deliberate vacations, but many not. There have been lots of them that have had large impacts on me. And people will say, which place had the most impact? And I say, I can answer that, but it's not a fair question, because the place that had the most impact on me was Southeast Asia. I wound up in Indonesia and then in Malaysia. And it was the first time I'd been in a non-Western culture. So it just had a huge impact on me. I just found everything stunningly interesting. And I walked around every day with my jaw hanging open. I was just like, whoa, look at that. But if I'd gone to someplace else that was a non-Western culture for the first time, that place would also almost certainly have made a big impact on me. So it's not entirely a fair question, but it's just different things. My wife and I went to the Galapagos one time, and that's a truly amazing place. And we went to Iceland, which is also a truly amazing place. But in Galapagos, it's the biology, and Iceland is the geology. So there's a number of places that had some very big impacts. But yeah, I worked in agriculture in Egypt, where we were up at southern end at a place called El Karg. You're at an oasis in the middle of the Sahara Desert, and they're growing stuff there. Yeah.
I was going to say, what has been maybe one of the most challenging places you've had to do field work? Was it Egypt?
The most challenging place I had to do fieldwork was Nigeria. The thing about Nigeria, you go there, it's a very culturally interesting place, and there's a lot to see. And I had a grad student who was working out of Ibadan, which there's a CGIR. It's a consultative group for agricultural international research. It's a big organization. They have a station there. And so he had field sites from southern Nigeria to northern Nigeria. It was extremely interesting. There's a lot of cultural diversity and all this kind of stuff. I read somewhere that a quarter of all Nigerians live in sub-Saharan Africa. So the competition for resources can be a little bit upfront sometimes. Yeah. An amazing place, but interesting in a lot of ways.
Sounds like you've had many fantastic travel opportunities over the course of your career. And you've been to, like you said, over 100 countries. I think that's absolutely remarkable. So are there any places that are on your bucket list that you haven't been to?
So I'm out about a year. So I'm supposed to go to Japan. I haven't been to Japan. Been in China when there was a lot of stuff going on with China, when China was starting to commercialize. So I was back and forth all the time. I was working with them. I think I went to China between 2002 and 2018. I think I went to China 20 odd times, but never went to Japan. It's like one of those things where, what, you haven't been to Japan? Go, yeah, I need to go. So I try to make it this year.
You need to make it happen.
I'm going to do the best I can. Yeah.
Well, enjoy your time when you're there. It's been wonderful to hear more about your research over the years, the lab, and the people that you've been able to work with. And I think there are still stereotypes in the media and just in the general public about what scientists are like and what life as a scientist is like. And in so many of the conversations I've had with scientists, it's clear that those stereotypes are not accurate, right? Scientists are these fun, amazing, creative, and funny people in many cases. Then you do a lot of cool things together on the research side, but also just getting together outside of the lab, making these quirky traditions. And I think there's just this human side of science that people don't always get to see. So we love sharing stories and just some examples of these quirky traditions or fun memories from scientists to kind of break some of these stereotypes. So Don, do you have a story of your own that you could share that shows this human side of science?
Well, 2 or 3 times a year, we'll go somewhere for an evening out. We just decide that things can get a little intense in the lab sometimes. You're working or all these pressure from granting agencies and things like this. And so we'll go out for a supper or something. And actually, we have a little lab funds that we can use to pay for that. We get some royalties on inventions and things like this. So we have a little bit of latitude to those things. Although I think actually mostly I pay for it. Anyway, my relationship with my students, especially as I've gotten older, they're more and more kind of my children in a way. So I'm always looking out for them. But yeah, we had a lot of interesting evenings away and things like this. And we were in Saint-Anne-de-Belle is the village. It's on the west end of Montreal Island, and it's where our campus of McGill is. The one student I have, she's very interested in all kinds of science. And we came out of the thing at the end and she goes, we should go down on the pier. I'm going, okay, why? Because there are 2 comets in the sky. So we went down and watched the comet. I mean, just nice stuff. It's always nice to see those things.
Well, I love this sentiment that you expressed of your lab feeling like a family. And I think that's something that can really help in terms of building that community and getting people engaged and motivated, even when times are tough in the lab. So I think that's fantastic. Do you have an example of maybe a fun field tradition that you do when you guys are out in the field, or just something funny or unexpected that happened during field work?
One of the interesting things, I have students from all over the world. So they're coming to Canada. And a lot of them, if they're from places near the tropics and things like this, they're always terrified about the winter.
True.
Yeah. So I had a student, she was from Kenya, and she was from the highlands in Kenya. So it was kind of weird because in the middle of summer, there was a group who was working in the field and she was experiencing temperatures higher than she had experienced before. And she was going, how can it be this hot and get cold? And I said, no, it's kind of a regular thing. Hot in the summer, cold in the winter, and it's continental climate here. So it's reasonably extreme, the differences. The next day she came to the field, she was going, well, how cold can it get? I said, well, the coldest it can get here is in the middle of winter night, things like this. It can get close to minus 40. Mm-hmm. So I was telling her that. And it was interesting because this is a person who she'd just come from Kenya. I mean, she'd only ever seen ice in a glass. And she looked at me and I tell it to her and I could see this has no meaning to her.
There's no barometer. Yep.
No. But the next day she came back out in the field again. And at some point she goes, oh, I was thinking about that minus 40. I go, yeah. So what do you think? She goes, it's like the inside of a deep freeze. I'm going, no.
It's worse.
Deep freeze is minus 20.
Yeah.
And there was this kind of pause, it must have gone on for like 20 or 30 seconds. And suddenly her eyebrows shot up and she goes, it's twice as cold as a deep freeze. I go, only at night sometimes.
So how did that first winter go with her?
Well, that's a good point because it's an interesting thing. They're always terrified of the cold. And I'm always telling them, don't worry about the cold. We have good clothing here. No problem. And no matter what the temperature is outside in the winter, inside, they're about 20, 21 degrees. I think it's about 72 Fahrenheit. But I always tell them, I said, what you guys need to watch out for is you're going to spend 4 or 5 months indoors. And the days are short. It's dark early. It's dark late in the morning. So when we had the first snow, she just thought it was magic. She was amazed. The first snowfall was just enraptured. It was really impressive to see. And by the end of February, she was clinically depressed.
Right.
So it's just the short days and living indoors. If you live somewhere where you can just step outside anytime, it feels very confining.
Yeah, I think you've got to find ways to step away, maybe get a little sunlight, even if you're standing by a window, things like that.
Yeah, skiing is good, but for students, downhill skiing, it's expensive.
Sure.
But just going for walks, even in the middle of winter, it helps. You just need to get outside sometimes. And if you go for a walk with some friends, then it becomes a social interaction. So.
Well, thank you for telling us more about your lab. And I think just some of the fun and quirky conversations you have with students really highlight this human side of science. And I think, like I said, that is really important to have this community because you're working together to solve some difficult challenges and problems in science. But oftentimes there are limitations, Don, whether it's funding or staff or technology or even just time. So if we took away the barriers that normally hold you back, what is the one research question that you would want to answer most?
What I'd like to do is what we're trying to do now, but on a much larger scale. I'd just like to know how much of this signaling is going on. The evidence we have between bacteria and plants, I think there's tons of it. I think it's got a whole series of motivations in terms of, evolution for the plant's benefit and things like this. It really does stimulate plant growth. And if they're stressed, especially, they grow better. There's tons of this stuff out there. And we dig up plants, I'll dig up different species and things like this, but we don't know. There's so little known about these things. If you look at what's known about the microbes that are associated with crop plants, it's some, but not a huge amount. I used to be kind of a botanist when I was young, so I'm always looking at the wild plants too. You look at what's known about the wild plants, there's much at all. So we've isolated some incredibly interesting strains off of wild plants. We isolated some off root nodules from the closest relative to soybean in this area, but it's slightly soybean-looking leaves, but it's small and things like this. And yeah, we got some strains out of it that were extremely interesting, one especially, and it's with the company now. So there's a lot of extremely interesting stuff in there. So it would be nice to try to get a sense of how much there is and how much of the signaling is going on. And I sense there's a lot of it, but it's not easy to make progress that quickly, or you end up writing grant proposals endlessly. So it's a major distraction after a while.
Definitely. And it sounds like just the scale of what we don't know yet in this particular area is massive. So you just need an enormous effort to get all that data collected and figure it out.
Well, actually, the first signaling stuff with the legumes was brand new with signals. And when we started doing the stuff with stress, we were the first ones to find that. And there's a lot more.
Yeah, I think it sounds like this is a hot area in the field where there's additional research being done, but always more work to do.
Yeah, I think that's right.
And we appreciate you sharing so many insights with us on our conversation today. I'd love to end by just talking about advice. So for listeners out there who may be on their own scientific journeys or otherwise, do you have a piece of advice that really helped you that you can share with them today?
In hindsight, it's kind of obvious, but I see people who are not necessarily following it. I mean, we talked about this already, but when you get a result that doesn't conform to what you expect, don't just brush it off. You need to make sure it's repeatable. But if it is, this could be more interesting than what you thought you were going to get. And it's always the problem with writing grant proposals. Most of the granting agencies are very good about that. They recognize that you're going to find things you didn't expect and things like this. Although it's interesting, I had a colleague of mine who was working with government people in Europe, and he was writing some sort of long-term 5-year thing. And they were asking him, well, what are you going to learn in this? What are you going to know? And actually a colleague of mine here in Canada, we got the same question. The person asked him, what are you going to know at the end of this work that you don't know now? He said, if I knew that, I wouldn't have to do work.
Right. It's a process. Yeah.
Yeah, that's right.
Well, I think that is a fantastic note. Is there any other last piece of advice or a last note of inspiration that you would like to leave our listeners with today?
Well, you have to persist. Things sometimes for reasons like we were just discussing, because things aren't exactly what you think. Things don't work out as you expected. But there's also, I don't know, I've got students who have personal problems and things like this. I had one a while ago. She was in the lab and she was a very good student. And then she came to see me because the country where she's from had broken down into a civil war. Things like that happen. And you just have to persist. There are any number of things that pop up and you just have to persist.
Well, I think that's great advice, both for science and life in general. We appreciate you sharing it with our listeners today. And if our listeners want to learn more about you, Don, and all of the amazing work that you've done, what is the best way for them to do so?
They could send me an email. My email address is donald.smith@mcgill.ca. But I should warn them, I get quite a few emails every day now. It's like 200 or 300. And so I try to get through all of them every day, but every once in a while I miss a block for reasons that I just get overwhelmed. So if you send me an email and I don't answer within 48 hours, just send it again. And I have a website at McGill, but honestly, I don't know what it is. But if you put Donald Smith at McGill, it'll come up.
Perfect. Well, Don, thank you so much for sharing your insights with us today. It's been such a pleasure to chat with you.
Well, thank you very much for being interested. I'm very grateful.
Well, it's been great to hear from you, listeners. Wonderful to have you here with us as well. We hope you'll join us again for another episode of People Behind the Science.